Okay, good morning. I think we can get it started because today we will cover quite a lot of topics. I'm not so sure I can finish it, but just give it a try. Also, I posted the last homework. It's a mini project to let you to use the DV analysis to find a new data set and play with this analysis. You can do this through the interface of Toolbox. So this lead you towards the final project. Your final project will be your presentation. Someone asked me, some of you asked me what's the presentation should look like. So it's open topic, anything you found is related to single cell data analysis. You can find the published data to do the reanalyze, or you want to use your own data set to apply some of what we learned from the course to your own data set, and get some preliminary results to report, to share with us in 10 minutes. Yes. I will give some more details on how we will score your presentation. Basically, we will look at the significance, innovation, approach, and your performance, how you deliver your talk. So you will get a spreadsheet, and with the scores for each of you, you will judge to each other. So, okay, in terms of the significance, whose talk will be number one, whose talk will be number two. Then for the innovation, whose talk will be number one. So we just rank each other. So those are the small details. So today we are going to cover several topics I put together. Not only the pseudo-time analysis, we will start from the batch effects correction. In terms of the technique, we will discuss CCA analysis, which one of the methods we can use this CCA approach to do the batch effects correction. And the more application is you use this for the multi-omics data integration. Then we will mention pseudo-time, or sometimes called trajectory analysis. So we will go through some principles behind each of applications. Many methods have been developed to do this pseudo-time trajectory analysis. So we will look at how they do it. So at the end, we will discuss a new multi-omics analysis, which is called the single-cell immunoraptor profiling. So this can be combined with a single-cell RNA-seq, but this is look at the surface immunocell surface receptor genotyping, basically. Okay, so we always observe this so-called batch effects. You assume if you do single-cell RNA-seq to the same population of samples. So once you embed the cells, if there's no batch effects, so you did same population, they all similar states, so those cells will be superimposed onto each other in this embedding low-dimensional figure. So that's if you see, oh, it's a clear separation, it could be the technical batch effects, and you want to correct for those. Well, on this example, people just say, okay, we have a control, and we have this treated with a NF beta treatment, those cells, they changed the position in the embedding. So clearly, there are red cell and those tail cell are in the different location. That's because some of this shift of the gene expression globally from one state to another. Frankly speaking, I still don't know why we need batch effect correction. Okay, but that's people want to see. Okay, we want to correct the batch effects. We believe this perturbation will have very minor or ignored, neglectable effects on the population of cells. So we hope the embedded results should look like something like this. Okay, so globally you shouldn't have this huge effects. I think that's a justification behind why people want to do this correction. I said I still don't know why, because what we normally follow, if you follow our strategy of this process of data, we will cluster cells into subclusters. So we will cluster this, right? Our strategy is over cluster cells. And then to annotate the cell type, if you have a T cell here, CD4 T cell here, and you also, so this is this example here, and you also found, okay, this is a T cell, CD4 T cell. It's automatically correct the batch because you will put them together by the name eventually. So I still don't understand why people want to do this correction. They properly will shift the cells from two different batches together, and then for what? For annotation. But this topic I have to cover it because there are so many methods have been developed. See, okay, we want to use MNN as a mutual near neighbor, and or this package is called the method called combat, and the Surat has its own pipeline procedure to do this correction. So people always say, okay, we want to compare and the different batch effects method, and we say which one gave you the best correction. So my concern, maybe because of my small knowledge, is why we need to do this. Turn out Dana Pierce, so one of these prestigious single-cell data scientists, she also mentioned in Twitter, now it's X, right? So if you click this link, you'll still be able to find this thread of, so batch effects is worst for cancer. So she's working in the cancer center. So she's doing a lot of cancer single-cell studies. Her argument is because each cancer is different and unique. Okay, so even you probably sequenced, sampled different subcolony of the solid cancer tissues from the same patient, you still see this separation of cell population maybe. So if you apply batch effects correction, it's always terrible. She mentioned in cancer, okay, but in general, I believe this applies to other cell types as well. So in her procedure, so they never do it, okay. Number of times I have to argue with the reviewer. So yes, it could be. It's a general belief that you need to do the batch effects correction before you can move on. So the reviewer just ask, okay, have you done the batch effects correction before you do anything? Anyway, through this, in front of even we discuss about the technology, okay, you can do it, but then you have to ask why is this, why you are doing it. Let's see what we can do. So, so the idea here is the batch effects present a technical variation. Okay, you cannot measure even the same cell to get the same results because of limitation of variation of technology. If that's the case, you want to correct these batch effects in order to allow them not confounded with the biological signals from single cell. You want to find the difference, the real difference between population of cells. So the linear method, which the combat has been here since the beginning of bulk RNA-seq. So people use combat to do the batch effects correction for bulk RNA-seq, even for the microarray study. But still, we can use this to do the single cell batch effect correction. But there also have nonlinear methods been introduced recently, including, so inside the Surat package, it's used CCA and some other specifically designed, so we will have a look of this. Just let you know so you don't be surprised when other people say, okay, I corrected the batch effects. So this is from a combat, the supervised batch effects correction using simple linear correction, okay? So that's an old school method. And most recently, so this is called most recently, 2018, use the idea about the nearest neighbor. So this is distance-based. You want to find your neighbors, you have to look at how far away from each other. So the idea is like here. So you have the baseline. So you have a batch 1, okay? Then in this case, you have X parameter, Y, and W, the 3 population of cells. Then you have a batch 2 has been embedded in a 3D disposition. So you shift the global of about all these batches. Now you look at each individual cells from here to find its nearest neighbor towards the different batches of cells. So you make a correction, and if you found, okay, the nearest neighbor of those population are matched in here, and this Y matched here. And also it have to be vice versa. Okay, the population cell you found their neighbors, this group, and this group from other way, so from this population, this batch, you look at back, you cannot say, okay, this population map to the nearest to here. So you have to be mutual. So this is called a neutral, nearest neighbor. So once you build those links, you merge them. Okay, so you put them together, which, like this yellow group, there's no correction, so they still have themselves as isolated new population. Unless you have another batch come in, you do the same thing. You merge by distance. This is very easy to understand, right? So it's based on embedding. So again, this is one thing I don't understand because you only move the cells, because here you have embedded cells. They all gave the coordinates in the embedding the space. So 2D, 3D, you can have a higher dimension, but you cannot see it. All you change is this location of cells. So you don't change the expression level. You don't change the original expression matrix. So UMI matrix still the same. So why you're moving cell around to cheat yourself. I still don't know why they do this. Okay, so CCA stands for canonical correlation analysis. It's used for batch effects correction. So you have one sample and then sequence them into different batches, so before and after treatment. And they found the unalignable, okay, unaligned this. So you want to redo this. You want to go through this CCA analysis to project them onto the same space, to allow them to share, to correlation between those projections as big as possible. So you found these corresponding positions, then realign them. So that's the involve two techniques. One is CCA, another is called dynamic time wrapping. So this is the same journal, same year. They published two technologies side by side. One is use nearest neighbor, another is based on the CCA. Same journal, same year, same issue. So CCA is not designed only for single cell data. It's used for correlating linear relationship between two multi-dimensional variables. Okay, variables means if you have a sample, there are many variables to align, to describe the samples. It can make, let's see, highlight this. It makes use of two views of the same semantic object to extract the representation of the semantics. So this is an abstract illustration of this two semantic view. So this is a view for the same data points, okay? So you see these data points, they are, each data points you have this population, they have different views. This is pink, this is blue, and we try to rotate this, okay? Same as PCA, you rotate on the rotation matrix, rotate your data points, and then you want you also rotate this, you found two rotation matrices at the same time. You want to end the products, they project onto the same space, then their correlation becomes the largest. To illustrate what the semantic, think about this. This is a database of pictures. So you have a lot of people's pictures, and in another database, you have their voice file, okay, the sound of this signal files. And this is a two view for the same group of people. So you want to align them if you have unlabeled. So you want to use this CCA projection to project the image into a space, project their corresponding voice into the space. They're totally different type of variables, but you try to maximize the correlation. In here you have pictures. It's one view. And then for the pictures, you have their text description. They're very, very different concept. Their variables and the types are very different, but you want to project these samples using the summarized list variables to rotate and put on the same space. So this is from a brain samples that you have EEG and also functional MRI for different views for the same object. You want to project those. Okay. Here illustrated you have found a function to project your x variable from view 1, and you found a function g to project the variable y from view 2 and put them together. So how this will be done? So you have red points, 3D example, and green points. Put them together, okay? Found rotation matrix, two rotation matrices. Project this and project that into a reduced dimension, and then do the regression to allow the correlation between the reduced or project points from these two views have a highest correlation between them. Okay, so in order to find this rotation matrix, we need information about relationship between data points or variables within this view and that view. So that's why in CCA we need a covariance between variables in first view, second view, so there's a variation, and use covariance between them. Then, this is a correlation, okay? That's a correlation after projection. So you project this, and then you try to maximize this correlation. So this is a mathematical expression of CCA procedure. So the goal is to maximize the correlation of the projected x and y. So you have A and B. So you can express the correlation between these two using that and this transform at the end into this form. Okay, C is a covariance matrix between variables in X and variables in Y. And then you have two rotation matrices, A and B, which are around it. So you want to maximize this whole thing at the end here. This is very similar as you do the PCA, that you want to maximize the covariance matrix, but that in that case is only one sample. So it's not between two samples. But here we need to do to work on this thing here, okay? To solve this, it involved SVD. Let's see. So this is a MATLAB function of the CCA analysis. You have X, Y, two matrices with the data you want to align to to each other. And then you found A and B, those are the rotation matrices, to rotate X and Y respectively. And this correlation you will get. So this is put CCA and the PCA together. There are some similarity between them. When we run the PCA, we only consider the covariance matrix with one view, because in that case, we only want to maximize the representation, the internal variability, to do the eigen decomposition using the covariance matrix from one sample. With CCA, you consider both, okay? You consider the correlation from variable within X and within the Y, and also consider the variation within the self as well. So we need to put this both A and B, these two matrices, to do this eigen decomposition. We can also rewrite this question as an optimization problem. So this is a lineage algebra solution. So you use SVD as to do this correlation, to find this rotation matrices. Well, you can also see, okay, I just want to make this as small as possible to get A and B, these two matrices. So this is another way to think about as alignment, because you found the distance after your projection of those points from X and Y views, and put them together and see, make them small, as small as possible. People also develop CCA algorithm applied to different situations. You have a kernel CCA, which you do the kernel transformation before you put them into CCA. Well, this is a conventional, and there are special treatment for the sparse data matrices, and this is also called a probabilistic CCA. So this is a kernel transformation, and then there's a directly linear CCA without the kernel transformation. There's just one more step. In some community ecology, there's another term. So it's also called CCA, but that's called canonical correspondence analysis. This is a very different approach. So although they all call the CCA, so you have to be careful when you talk about the CCA. In our case, we are talking about this correlation analysis. Well, the correspondence analysis is a very, very different approach. So just don't get confused sometimes. Then there are a different technology called dynamic time wrapping. So this is allow you to optimize a match between two given sequences. You have two sequence data, like samples. You get a signal from different time points, but you sampled in different frequencies. So one sequence maybe have a longer sequence where another is not. So you want to align them by the distance to allow them to align, okay? To introduce the space between between sequences to make them matched. Okay, so what's the matched? So if you look at this blue curve, after this dynamic time warping, it becomes stretched and matched with this orange curve. All right, so that's the technique we will use to finally align these trajectories. So this is a MATLAB function, dtw, and this x, y are two sequences of data. This will be distance after align. Then you will get the new positions of those coordinates, so we can play out, get the results very easily. So within this algorithm, we compute the pairwise distance between sequence A and B. So these two sequences have a different length, okay? Then we compute the distance and then try to find a path which allow them to align, and so this is the way to compute. Okay, so we now jump into trajectory analysis. So the idea here is cellular biologists always want to track the change of the cell states and also follow their development or differentiation stage. And if you can track individual cells' fate, for example, in study embryonic development, or you want to study the turnout of tissues, homeostasis, stem cell differentiation or regeneration into other cell types. So we want to follow the trajectory of individual cells. So you can have some marker-based, image-based, probe-based, using fluorescent, for example, to follow those. You can use mutations. Okay, you can detect some of those. There are many lineage tracing technique has been developed. But with the single cell data, we try to make this easier, because this design, those technique really take efforts. Not so simple, not every lab can do it. But our idea is to assume the populations of cells you collected, they are representative of cells in different states. So maybe we can, through the computational method, to rearrange cells and follow their development trajectory or differentiation trajectory to reconstruct time points of the single cell which developed at a different time. So this idea is not new. It had been there from the very beginning. Even the 10x genomics technology become available, people already start to think about it in this direction. And we also know this cellular process is not purely random. You have some kind of a cannibalization. You follow the trajectory of the development control genetically. You only have several possible paths rather than purely randomly wangling around the space. You have this kind of, so the cell differentiation will follow several trajectories fixed without even be bothered by the randomness in the environment factors. So it's a very robust process. So people believe this cell line or cell trajectory, the can be learned from observed individual states follow this trajectory if you have enough samples along this way. And those space are the empty space where no cell states will explore those space. So that's what people generally believe that the cellular dynamics is follow this landscape of the canalization. So the purpose is to model the transition, okay, to cell identities or cell states, to model or to simulate the differentiation procedures. And after you build these trajectories, you can start to look at the genes, see which one follow this, which one against that, which one you change the expression dramatically in the middle of the trajectory. So you can have some established hypothesis for understanding the cellular process. So once you have those knowledge, you build a hypothesis, you can test them in the lab. Any questions for this part? Okay, so this is not too hard to understand. If you have multiple samples in this different colors, okay, then you can put an early stage of cells you collected from this sample 1 in here, and you know those are the sample 2, then you have time. But if you don't have this information, you just collect a group of cells from same time point, a population of cells, you want to have their markers have been labeled, and then use these labels to reconstruct this time trajectory, okay? Then let's infer the change of the genes levels, see whether gene A or B are associated or are the driver for this process. You can have a branch, so if you have in the early stage stem cells, for example, you can move from one direction and then further differentiation for the branches. So in that case, you are not only build one trajectory, and you can have multiple pseudo-time trajectories from one population of cells. So this is one of the earliest studies using this method, and they found pseudo-time and the gene associated, and this is using stem cells, embryo stem cells, to derive different lineages of endothelium cell, okay? I think in this study, they found there are two different pathways you can achieve similar results at the end. And they follow, they use experimental approach to control the cell, go this direction, go that direction, and follow the change of the expression of the genes. Another illustration, so I keep a lot of those to make you conceptually appreciate this method. So if you think about this picture, it has to be taken across the time points. But in single cell data study, we just have this picture, okay? This picture is not, it's taken, we have a snapshot from single cell data. It's just one time, but it contains information which help you to build a trajectory. Another example, I try to explain this to people outside the single cell world, how this works. You don't have a video camera, you cannot take serious picture for the movement of this horse in order to study its gaits. But if you have a picture, a static picture, just one of them, but if you have a lot of the horses here, you can rearrange individual horses and to regenerate this series of movement, the gaits in a different position, and probably can reconstruct this, the movement of a typical horse. So here is we have t-SNE embedding for single cell data. This is for the t-SNE is for the purpose of the clustering. So you want to emphasize their different isolated subclusters and to do the cell type annotation. But if you believe they are a series of cells, they are related by the development stage or their change from one phase to another. So you can find a way to do the different ways to do this embedding. In this case, we emphasize the change of the cell states from one to another and form this kind of linear shape rather than emphasize them into subclusters. So you change the embedding method and you believe their underlying biology is true, then you can start to think about this population is either start from this end or start this from this end, or whether there's a branch. Okay, so it's based on your understanding of your cell type. So if the signal is strong, you can use very simple approach. So PCA even can do it. So this is a very strong signal. You found the change of states of cells from one to another. People also want to build a network between subclusters of the cell population, and then to see whether there's the trajectory goes through this network and see which one makes sense. So this is 2017. It's a very early stage of studies. So these are the selection of several pseudotime analysis tools: Monocle and Slice, T-Scan, SCARP. So each of them use different technology to do the dimensionality reduction. Use ICA, PCA, or t-SNE. Okay, they all have the dimensionality reduction steps as first. They project the cells and then start to, this is called the minimum span tree construction. So after dimensionality reduction, you link the cells with, make a network, okay? You connect the cells. And then found a representation of a network. And then from network, you found the pathway, the tree from the this path, okay? There are different ways. In this case, you build network between cells. You can, this is from the cluster centers. This also uses cluster centers. This method uses a curve fitting. So not to build a tree, but it just found a curve, goes through majority of cells. There are different computational techniques you can use. No one can see, okay, which method is correct, as long as you build a trajectory, and it really depends on the downstream biologies and interpret the results. This is explain the minimum span tree. So WeView found, so what it is. So we will find the shortest paths, okay, minimum combinations. So you can connect all the dots in the network using this tree structure. So in this example, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 nodes. We have 10 nodes, and they all linked, and the distance between them has been labeled with numbers. So what we want to find is combinations of links, and these links will cover all the nodes, okay, to make, so you can through this parse to visit all the nodes in the network. But you want to have this total weight or total path as the shortest as possible, the combinations. So you want to find at the end this 3 plus 1 plus 4 plus 7 plus 8. The total you put together is the smallest you can have to have this tree to cover all the nodes. Okay, so this is the algorithm. Yeah, so the application for this minimum span tree is you can see that on all these three methods. So these three methods are all based on the network between cells. You can either build a network for all the cells, or you can use representative cluster center of cells. Anyway, so you put the cells together and you make a link between cells, you compute the distance between cells. Then you put your minimum span tree onto it. Okay, after that, then you can start to follow the minimum span trees to build the trajectory. So once you put the minimum span tree, you can start to from somewhere, and then from here, for example, you go to here to build one trajectory, and then you can go from here to build another. Okay. Another algorithm also the graph-based. So this is used in these two methods, Wishbone, Wanderlust. So these two methods will build a nearest neighbor graph. Basically, it's build a network. This network is not fully connected. For example, there's no link between this to that. It was a fully connected network, but it's been trimmed by the nearest neighbor constraint, and you make a nearest neighbor network. So this gave you some local connections. If you're close to each other, those nodes will link those cells, and then when you move around away from this, you start to build next neighbors. At the end, you end up with this kind of network, and then from here to facilitate you build a shortest work, or you build a curve go through this. All right, so once you have the nearest neighbor network, it's relatively easy for you to start to find the path from one end goes to another, follow this local trajectory. So this is example of nearest graph. So I don't know if I can run. So this is a small Java-based online demonstration to build nearest neighbor network. It shows you when you select different k and you get different results. So you can simulate the number of points. So you randomly generate random number of points, then so depends on, so let me generate more points. So it depends on how you decide as a nearest neighbor. So if you select each one, each node, you will find 5 nearest neighbors and make link, it will generate a picture like this. If you only allow to have 3 neighbors, then it generates a graph which only considers 3 nearest neighbors. So slightly sparse network. So if you increase this to 8, the network becomes more busy or more connected. So this is example when you have a k equals 3, and this becomes separated to isolated subnetwork. But if you increase k to 4, and this becomes linked, and this whole cell network become more connected. Okay. This is in MATLAB. We can compute network cells, and this is in the t-SNE. Then you select the k, and then create this k nearest neighbor network. And from here, we can build a trajectory. And then once you have this, you can find the genes which either increased with a trajectory from one end to another, or downregulated those genes. So this function is available in the toolbox. There are other algorithms. Okay, so this is based on cluster network. So this connecting of cluster centers. They're cluster triplets correction. But anyways, people from data science just try to use all different kinds of methods they can have, developed many of these trajectory build methods. I also want to mention the RNA velocity. I don't know if you heard about this. So this is use information from splicing and unspliced mRNA counts. So in this RNA velocity method, we build 2 matrices. So this is a little bit different from building a trajectory. So this is use different information. So I want to mention this RNA. So from single-cell RNA-seq data, we will get 2 matrices. Normally, we get a gene by cell. So for this analysis, we will separate this count matrix into 2 matrices. So when you sum it together, you add them up. This will be the number of gene expression for this cell, this gene. So this could be 10, this could be 25. So you have numbers here. But in this analysis, it requires you to separate this into the counts for the spliced, unspliced. mRNA counts. So this 10 will be separated into maybe 5, 5, okay? Means this gene in this cell, number 1, their half of their counts from unspliced mRNA, half from spliced. This 25 could be changed to 20 and 5. So you get 2 matrices. So this ratio, 5, 5, 20 versus 5, to indicate the splicing states of the cell. If you have actively transcribed cells, genes, then this gene should have a lot of unspliced, because you haven't have the time to do this. How to see? Cut the introns to merge and put the extron together. But if you have more mature, stable expression of this gene, you are not constantly provide unspliced new mRNA in, you will have a ratio which is different. You will have a more spliced and less unspliced. So this method just use that information to build the dynamic between the balanced, spliced, unspliced, to use that information to indicate the cell states. So to put the position of cells in, depends on their transcription states of the balance. Okay, on this slide we keep mention in a little bit of detail about this package called monoclonal, because monoclonal is one of the most popular options in single cell pseudotime trajectory analysis. However, monoclonal is not a very stable package. It has been changed from version 1, version 2 to version 3 right now. The developer may be working on the version 4. And also each version, they use different algorithm. Although it's called the same name, so if you use monoclonal 2 and 3, you have to mention which version you are using, because the underlying algorithm is very different. So the original version, it started from 2014, so this is really, really early stage. They use ICA to reduce the dimension, and they build a minimum span tree on it, then to build the trajectory. They work on this proliferating cells and differentiating cells in the different stage. In monoclonal 2, they changed the embedding algorithm to reversed graph embedding. I don't know what it is. Claim the version 2 will produce more accurate, robust, require no prior knowledge. So there are some of this explanation of what is reverse graph embedding. So they use this to learn so-called principal, the parsimony principal graph, basically is a curve. This curve will pass through the middle of data points, and also allow them to have a branch. So this is a principal curve goes through and allow to have a branch to make a graph, basically a network. So this is a Protocol 2, and you'll use DDR tree, whatever. So this is the process of this. Okay. And the dimensionality reduction technique, and then build the tree with a branch. So this is network. Select the root. And then this is Protocol 3. Protocol 3 try to be not only work on the trajectory, but also have a function for the clustering and also differential expression. And use cluster, use UMAP and the Louvain community detection technique to do this. But still, people still only use Protocol to do the pseudo-time analysis because of historical reason. Nobody use Protocol to do clustering or differentiation analysis. So I don't see many of the applications here. People still using Protocol because that's the name of the brand of this package. So how the Protocol 3 is working, let's see. I don't have a slide on here exactly what's inside the Protocol 3. And not too much different from Protocol 2, I believe. In the toolbox, I have this function you can call Protocol 3, but that's in R, so we have to install R, and then from the MATLAB interface to call the R package to help you to build the trajectory. And in the middle, you need to select where you believe to be the root, because when you have the network, you want to find the trajectory, you need to indicate where do you believe is the beginning of the trajectory. So after that, the package will automatically build those trajectory for you and give you the pseudo time. So we will play with this algorithm in the computer session, so you know how to use this. Okay, it's still the most popular one recently. Slingshot is another one based on the principal curve. Sum this in the middle, you know this minimum span tree. This is a review paper summarize existing pseudo-time methods and then show different dimensionality reduction, whether it's based on clusters or the cells, what kind of graph used internally, how to compute, whether allow the branch, supervised need tell the program where the starting cluster or starting cell is. Slingshot is another package claimed to be robust against several others. So it's also it allow you to build more trajectories. This recovers 3 of the paths. Well, the other method may not be able to recover all the trajectories. So multi-linear inference. TS scan, another method, and well documented. I could re-implement the whole thing by just reading the paper. It's pretty good. Stream, another one. Lisa is one of my someone I know before. They developed their own, but not very popular. So this is a review paper, put everything together. You can see a large numbers of different methods. So this is like systemically has been benchmarked and see which one is good in which situation. The overall score accuracy, scalability, stability, usability, etc., etc. Let's see the time. Let's take a 5 minutes break. Okay, come back 11:15. Loading, okay. Okay, so let's continue with this evaluation study for pseudo-time analysis methods. So this table just continues. I cannot put on even one slide. We have to cut into half. So this is a really comprehensive review about all these methods. So this also tells you there's no converged consensus of which analysis you should use. You can use any of them because they are published, and review won't be able to judge, say, okay, you use the wrong method. They're probably not, because these methods are all developed for trajectory analysis, but they definitely will give you the different pseudo-time assignment for your cells. So I tell you what I'm going to use. I use, we will do this Thursday in computer. I use a FATE. FATE is one of the dimensionality reduction algorithm. So it's in parallel with the UMAP and the t-SNE. UMAP and the t-SNE are in the same category. It's regenerative model. It's populate up those cell clusters by whether they are the probability they are neighbors in the high dimensional space. So this location of those subclusters by this t-SNE UMAP are not fixed. They are randomly populated. So this population, when you run algorithm, it could be here, and next time it could be regenerated in this location here. Because those algorithms contains this regenerative model to just put cell populations in the space somewhere. So you should not use this as embedding algorithm to do your trajectory analysis, because each time you have different placement, different location of your population of cells. So FATE is based on MDS, multidimensional scaling. So this keeps the original location of the map. Remember the distance matrix between different cities? You want to use MDS to reconstruct the map, which keep the location and the distance between all the cities as much as possible. So this algorithm allows you to keep the relative distance between cells, not lost the relative position globally. But here, t-SNE UMAP, you lost those real distance information globally. You just try to generate isolated population clusters. Emphasize on the neighbors, okay? But between neighbors, there's no link. But the FATE is okay. So I use FATE to project cells, and then I use manual drawing. I don't use any algorithm. I just draw the line. That's my result. Okay. Well, it's very difficult to see, okay, which method is good, which method is bad. Quality, paper, so whatever. But I found it's headache to look at this picture. This is continue and the same figure 3, and yeah, so number 1. So this should be the best, I believe is, well, I don't know which one. Let's see Monaco. So the Monaco ICA, this is Monaco 1. So this should be Monaco 2 based on the DDR tree. Okay. So based on this analysis, they gave you some guidelines. So which method you should pick, what you should consider. So I just pay attention to Monaco 2, this DDR tree. I think Monaco 3 has not been published yet. Yeah, but this should be the algorithm still used. Anyway. So this is also, it's a R package, try to put all the pseudo-time data analysis method together under the same umbrella, including some downstream analysis. For example, you can find the change of genes. Okay, so follow the trajectory, early stage, later stage, whether there are major change between the genes, between the older or younger or upstream, downstream gene cells, whether their gene expression change. So they try to build those. Well, the real biologists still emphasize like another Twitter citation, which is claim that the pseudo time is not actually time. So it's just a lot of the pseudo claim. If you believe it's a strong, believe it. So that's what something I was considering to do. We try to follow the trajectory if you build, and then using sliding windows to follow the trajectory to sample the cells. So if you look at it here, this is a window 1 from early stage, then you follow the trajectory, have a window 2. The cells located within this window will be used as sample 1, sample 2, sample 3. Then for each sample, I can build a gene regulatory network. So this is 1, this is 2. So follow the time trajectory, we build the networks. Then there are some analysis which you can have array of networks and work on this as a tensor. We will discuss tensor and other structures, networks next week. So there's some analysis to learn the major components within this multiple network structure, and then see whether there are any major components. Remember the idea about components, okay? So you looks very messy, but if you follow some major meta signals, you may reconstruct some of these components through network change and try to learn from there. So that's some idea. We try to use information which can only be reconstructed from the pseudo time. If you don't have the pseudo time, you don't have this time trajectory variable to play with your data. So I think that's adding values to study the single cell data. So this is a real example illustration. We have mouse data. If you extract the neurons and build the trajectories, this could be neurons in the different layers of the brain. okay? So this may be not a pseudo time. It could be the anatomic structure of the different layers of the brain and have a different subtype of the neurons. But same idea, you can apply this. Again, here I use a fate 3, so 3D fate, so it makes sense to have this trajectory. Otherwise, as I said, if you see someone use UMAP t-SNE to build a trajectory, you know they are wrong, okay? You can stand up to criticize. So RNA velocity, I just described here. Use the information from a spliced, unspliced ratio between cells, between genes. So this is built on this understanding of this dynamic of gene transcription. So if you have a gene in the DNA on the chromosome, you start to transcribe, build pre-mRNAs, and then you through this mRNA processing to make mature RNA, you cut and then also merge those, so the spliced become spliced. So if this process is continuous and stable, then you should have a balance between the ratio between the concentration or count number of spliced and unspliced counts, okay? So this gene, if it's balanced, it will be located on this diagonal line. If you change the parameter a little bit, adjust a little bit, so you should allow the genes to project onto this straight line here. If you found a lot of this unspliced, that means this has been upregulated very quickly, rapidly. Transcription has been upregulated. If you go down, see, okay, that means some factor has stopped the transcription and then allow you to relatively accumulate a more spliced version of this gene. So where this gene is located tells you the change of their balance between spliced and unspliced, and indicate whether this transcription has been upregulated or downregulated. So you can do this for the gene for all the cells. So if you have this spliced, unspliced counts, and then for one of the genes, you can put, okay, for cell number 1 is here, but another cell is here, another cell here. So you can put your gene, your cells in this space. If majority of cells is located in this place, like where this place, for that population of cells, for that given gene, it's upregulating that gene, okay? Because of the shift of the location of this distribution. I think they use a very strong indication, which is a circadianism related gene. So they know this gene follow the trajectory of the time. In the 24 hours, this gene should be up and down, up and down. So this is a very strong pattern. So if you look at these genes, spliced, unspliced ratio changed, follow the time. So they can find those genes and map those genes into this scatter plot, and they know, okay, when this gene has been upregulated in the early morning, for example, it starts to show up in this corner of the scatter plot. Then when you move, this gene start to slow down transcription, they start to move to this corner of the this scatter plot. All right, I hope you get the idea about this. And it's a very, very powerful approach. So this is gene is FGF1, okay? And this gene, what they found is a CBS. So they have different phase, okay? Phase means the peak expression of the two genes are at a different time. I hope you get the idea about the RNA velocity analysis. We still have 30 minutes. I will finish off with this single cell immunorepertoire analysis. I don't know how many of you will have a chance to use this, but it's a very, very important, very powerful approach for immuno cell analysis in the immunity study. So the idea is every T cell, B cell, which have the receptors on the surface. So for the T cell, it's TCR, T cell receptor. For the B cell, they're BCR. So BCR is try to respond to generate antibody, so B cells, and it depends on the antigen. Well, the T cells will use this receptor to communicate with other macrophages, for example. So those are the very important to cross-talk the signals on the surface of T cell and the B cell immunocells. So this is a cartoon to illustrate communications between T cell using this is helper T cells, one type of the T cell to facilitate those communication. Use T cell receptor with the help of CD4 receptor together and make a complex, and this will be communicating with a dendritic cell and also have the receptor presenting molecule. And dendritic cell will have a contact with pathogens, for example. Well, this T cell receptor shows strong level of this diversity. That means they all encoded with a different subgenes or subtranscriptions in the different ways of combinations. So this receptor has V domain, J domain, and a different type of alpha chain, beta chain. So, so those gene clusters will randomly be transcribed and encode the receptor subdomains. So you create this different combinations through gene rearrangement. So each T cell on the surface, because of gene expression difference between this, these combinations give you the diversity of the receptor. So if you have alpha beta type of these combinations, the major subclusters, then you will pick from so V segment for the subdomain, subunit of the receptor. You will pick out of 7, you pick 1 randomly. Then from the J segment, there are 61 different genes encoded for this, so you can pick one of them. So this is a combination. Then you have a beta chain as well. There are 67 different selections and 30 different, and also you have multiple subdomains in their combinations. So this is create a huge possible combinations of those subunits. So this is a place of the V domain. So this is T cell receptor beta chain V domain, okay, the cluster and the different subtypes. You see there a lot have been arranged on the chromosome. This is in the chromosome number 7, the human, in the human, yes. So your T cell receptor will pick one of them by random to present and express that particular one in its surface. You change another T cell, you may have this rather than that. Well, when you change another T cell, it will have this version rather than the other two. So every time you have a different combinations. So this is a T cell receptor, the J, another region. So this is also in the chromosome 7, another 14. So those are the clusters of those subunits. So this is a rearrangement. When the T cells start to express this, the whole thing, through the rearrangement, it start to pick one of the V and pick J, one of the J combined, another subdomain, then put together. So this will be the surface receptor, T cell surface receptor, with this multi-domain combinations through this rearrangement. So for this rearrangement, they gave you 10 to the 14 power combinations, and then become this is dimer. So this you have an alpha and the beta. Together, this gave you all the surface possible combinations of the receptor, 10 to the 21, 10 to the power of 21s, pairs of the possibilities. So this large diversity gave you the flexibility to respond to different receptor, to different external pathogens, other similars. Depends on which colony will be selected. Imagine you have all these T cells with a different type of this surface receptor. And some may be very useful to fight against viral infections, some may be against bacterial infection. So when you have flu and this viral infection, then this subpopulation of T cell will get amplified. So you have this particular combinations among such many of the diversity will be overemphasized, amplified. So you'll start to have a lot of those colonies show that combinations of receptor in their population. So 10x Genomics developed this new kit called BCR amplification and the TCR amplification kit. So this kit will be emphasized to amplify the region which to sequence the receptor region. So allow you to have a resolution to tell which colony and which type of the receptor has of that colony has been amplified or has been over-represented in your single cell data. Mainly focus on the T cell and the B cell. Okay, so this is similar. In the B cell, you also have this light chain and the heavy chain, the combinations. Well, most of the T cell have alpha, beta chains, for example. So this kit will label the cell in the same way we do single cell RNA-seq. So the first step is the same. Then, so this is the standard. Yeah, so this is all the standard procedure we have already described in single cell RNA-seq, regular. But now, once we prepared this, we start to have this bar-coded cDNA. Then we put this half into regular. This is a regular single cell RNA-seq. Single cell RNA. So this is what we are doing in routine. But it will put another half into here, okay, to specifically amplify the T cell receptor region. Okay, so this will be receptor region amplification. If we are not doing this repertoire analysis, we will go through regular single cell RNA-seq. That's what we normally do. We label the cell and then start to sequencing this library. But now this library will be put half into T cell receptor. In this side, there's a new pairs of PCR primers. This PCR primers has been designed just amplified this particular given target region. This region contains the VDJ segments. So depends on which template that T cell provide to the kit, it will amplify that region. So at the end, we will get side by side two sets of data. One is regular single cell RNA-seq data. In there, you can see, okay, I have 50% T cells in there. I have 20% B cell in there, and some other cells. Well, for each T cell, B cell, in this side, you will have their receptor isotype, basically. You have those sequence, and you know which combinations of those VDJ segments are. So this is two libraries have been constructed. This is a regular single cell RNA-seq, and this is a enriched VDJ segments enriched library. So in the region of those amplified region, they contain different information. So here is this tells you where this gene is transcribed. So this gave you the count of genes. Here gave you the count of different type of the what the sequence of this region. It's not a count, it's just a sequence. downstream process also different. You know the cell ranger, if you have this repertoire analysis, you generate the data from this VDJ receptor sequence enriched. Then you have to use cell ranger VDJ specific command line. And this run will give you a different results, which is you don't normally see from the typical single-cell RNA-seq expression results. They have a colony type. They have reference, so those cells, those extra files. When you open this, all contig annotation, so this is only produced by this VDJ analysis. You found this particular cell. So the barcode is the cell. Okay, most of them should be the T cell, B cell. Then you should have, for this particular cell and what the sequence, basically the contig tells you sequence of that receptor region. Receptor region you should find, you use this, you can find another file, we will have the full sequence. But here just tell you what gene in there, so what type of V genes. So this cell have a T cell receptor alpha chain V3, so there's a V and there's no D. What's the J? So it's receptor AJ27. Then there are CDR region as well. So this is a full sequence we get from the TCR region for this particular cell. And then you move to another T cell, you have different combinations of this. Then you just move on. Okay. So this is a place you can look at this contig name and find its full sequence. So the colony type, you can find the sequence of the alpha chain, the beta chain, and this sequence can tell you this will be alpha chain V3. This region could be J27, something like this. So you have full sequence information for that particular receptor region and tell you this T cell is amplified, this particular type of the T cell receptor on the surface. You will find a lot of redundancy at the end. So all this, for example, are T cells. They have a lot, but all these blue ones share the same consensus. What that means is their surface receptor sequence are the same. They all amplified from one T cell. They get many copies of this T cell because it's likely to be there has been unproportionately amplified due to requirement to fight some conditions. Okay, so if you have unstimulated samples, all the T cells may be on its nature states. There's no particular colony is amplified. So you see all different kinds of receptors. But in your sample, if you have something like this, 70–80%, all the T cells share the same receptors. You know they start from the same cell, get amplified because of this particular condition, require you have a T cell to generate a lot of those stimuli. So by collapse the information from the VDJ, you can find the frequency of the colonies. So this is one type. This another. This another. So you have some idea about the colony size distribution. So this is using the Looper browser, which is a built-in the 10x genomics provided pipeline. Yeah, I think that's all. I hope you get a basic idea of what we are trying to do. We try to follow the copy numbers of particular given or T cell receptor isotype to see whether they biased towards particular receptor type, and then to infer, okay, this particular T cell as a population is under some of the conditions which allow this particular given type to be amplified. You may find from different studies same receptor types, maybe like this one, always get amplified. Then you have some idea about this type of the receptor may be specifically useful for fight this kind of disease, either it's a cancer or infectious disease or other conditions. Okay. Any other questions? We still have several minutes, so you have a question about homework. Remember, you have a homework, the last one. Yeah. Oh, you can if you want. If you run out of samples and you want to say, okay, my mini project homework number 4, the slides I can use into my presentation is fine. Remember, the presentation is between you. You judge your peers. And so who's the number 1? Who's the number 2? So number 1 get a 10 score, and number 2 get 9, number 3 get something like that, okay? You judge between yourself. You also vote for yourself as well, so you may want to put all the category for yourself as 1. It's okay, yeah, but I take average at the end. Okay, you know, you got the idea? So we judged by your presentation by significance of the topic you selected to discuss. Innovation, so whether you build in some of these creative ideas in your project. Approach, the methods you use is reasonable and will help you to get the results which make sense. And the performance. Performance is your oral presentation, your answer questions, whether in a good way or not. So the performance is, so 4 categories, and you get a rank between yourself, among yourself, for each category. Okay, so you will be here. We will be in this room, and we will sit. Okay. Okay. Yeah. So everybody will have a total 10 minutes. So you can decide, I talk 8 minutes and give 2 minutes for the questions, or you want to talk about the 7 minutes, give 3 minutes discussion. It's up to you. But that will count into your performance. Okay. Sarah will be here listening to. No. Yeah, you can. Yeah, you can work. We tried. It's pretty good. It worked very well. Mm-hmm. Yeah. I think so because the whole course is a single cell based. Yeah, yeah. You have to have some single cell data in there maybe. Yeah. You can just like the mini project homework, you download somewhere, and you just assume if I have that data from that tissue which relevant to my research, I will do this. I already done this and found something interesting. Yeah. Okay. Yeah. Hope you can find some creative projects, idea. Okay. Mm-hmm.
Last time we were talking about ligand receptor pair and the score to sort of find the ligand, the unknown ligand for copper hepatitis. And later I figured out that the receptor is actually mainly regulated post-transcriptionally. So it would be like endocytose and then degrade, but the mRNA expression is still the same. So with that, I think that would bias the ligand receptor score a lot. But is there a way to bypass this and just go from the ligand to the differentiated expression?
Oh, I never thought about this. So you have so this is some real data you play with?
Oh, no, this is just the
example.
The structure of the biology underneath copper metabolism. So normally there's a ligand, and for iron it's hepcidin. So a lot of literature have suspected that there's a ligand that would sort of downregulate the expression of copper transporter 1, which is the main copper transporter, and is responsible for shuttling copper inside of the cell.
Yeah.
And then after copper is shuttled inside of the cell, it would then activate some copper-associated transcription factors.
I see. Yeah.
Yeah, but because this, the receptor, that we think that the ligand has that effect too, so it downregulates it. So they should be negatively correlated. Is regulated mainly post-transcription.
Oh, I see.
So we were wondering if there's a way to use differentiated gene pathways to then come back to predict the ligand, or would that be really biased because the pathways may also be affected by some other intracellular signals?
Yeah, I think you're talking about, so this is also across different cell type.
Oh, so
no, this is ligand from environment.
We think the ligand is also from liver.
Uh-huh.
The